Hi class, and welcome to your next video cast on the domains Archaebacteria and Eubacteria. Remember we talked about when we did classification, domain was a new type of classification, and now we're going to deal with bacteria, which are going to be our prokaryotes. So remember back to when we did cells and we talked about prokaryotes, that's what we're going to use now. So let's go ahead and get started. So this is just a photo of a red blood cell and some bacteria. Now we can see that it's gram-positive because of its color. It's almost purple in color. And that gram-positive has these, what are cocci pairs. They're circular in shape. And that makes us think that it is Streptococcus. The same type of bacteria that's going to be present in your throat when you have strep throat. That's why we call it strep throat. So we have characteristics of bacteria, and bacteria are generally called prokaryotes. Remember, back from when we did cells, they lack a cell nucleus and other membranes around membrane-bound organelles. Now, remember, the one organelle that they do have, since it is not membrane-bound, is a ribosome. They are smaller than eukaryotic cells. A lot of times, we don't notice that as easily because we see two different pictures lined up next to each other and they seem the same size, but they are actually smaller. And then they're divided into two kingdoms, Archaebacteria and Eubacteria. So, and then like I talked about earlier, in the 1990s we elevated these to domain status due to their DNA sequencing. Now remember, that's part of evolution. So based on our evolutionary tree, we're seeing that these are further apart from the eukaryotes. So we want to distinguish that in our taxonomy. Okay, so here is a picture of a bacteria cell, and this is just your model bacteria cell. By no means does this mean everything looks like this. So if you look in the center here, we have that bluish spaghetti looking stuff. And that's going to be our nucleoid region. Remember, prokaryotes only have one chromosome, and that one chromosome is a circular chromosome. It is not enclosed like in eukaryotes. It's just sitting out there in the cytoplasm. And then around here, these greenish looking things are your ribosomes, where we're going to carry out protein synthesis. Remember, protein synthesis needs to occur in any cell for it to survive. Now, it is then surrounded by your cytoplasmic membrane, and you also have a cell wall here. The cell wall is generally made of peptidoglycan. I'll say that one more time. The cell wall is generally made of peptidoglycan. It's kind of a hard word to spell, but I'll go ahead and spell it up here for you. So we also, encapsulating the cell wall, have what is called a capsule, and that's where we're going to find other structures. They're called our pili, and the pili help us kind of move around. They're kind of our sensors. They feel what's out there. Now, there are no eyes. There's nothing to guide it, so these pili are kind of just like sensing where we should go. They do sense other things besides like light and solid. They sense pH, they sense moisture, so they are very important to our environment and to our bacteria. The other structure you might notice is a flagella. That's the whip-like structure that helps it move in a whip-like fashion. Now, we can have flagella, or we can have cilium. And cilium, or plural cilia, just kind of whip back and forth, and they help us move help that bacteria move as well. So you can have either of those. That's very common. Okay, so let's just kind of mosey on along now. We're going to talk about the structure of the bacterial cell. Now, the structure of the bacterial cell has a capsule with pili. Remember, I just talked about those pili, and they're on the outermost layer of the cell. And that's for protection from drying out, being eaten. And remember, those pili just kind of help us sense things. The pili are hair-like to help attach to surfaces as well. So it's going to help a lot in movement. We have a cell wall made of many, many molecules called peptidoglycan. And we have a plasma membrane. Remember back when we did cells, that plasma membrane is going to be the same. It's got a phospholipid bilayer, so you have those phospholipids that have the phosphate head and two fatty acids. One of them is going to be unsaturated. And this is where cellular respiration is going to take place. Now, we're not going to fully go through cellular respiration like we learned about earlier this year. What we're going to do is we're solely going to do glycolysis. So remember, glyco means sugar and lysis means breaking. We're going to break that sugar apart, getting about 2 ATP, and that is how we're going to go through cellular respiration. We are not going to go through the full part of cellular respiration because we don't have any mitochondria. So, more structures in our cell. Obviously, we have cytoplasm. That's just the fluid-like part that holds it together. We have the nuclear region, like I talked about. That is where we find the circular DNA. We have ribosomes, which we use to make proteins. And finally, we have a flagella, or we can have cilia. Now, we have these two different types of bacteria, and I've mentioned them a couple times, but I haven't really described what they are. So, archaebacteria, archaea meaning odor, is the odor bacteria. They are actually extremophiles, meaning they live in extreme conditions. So, they really like those extreme conditions. Their RNA sequences are found to be very different from eubacteria. This is why we split up the kingdom bacteria. And now we have eubacteria, archaebacteria. And actually in these, we have no peptidoglycan in our cell walls. So that's going to be pretty important because that's going to help us distinguish whether we have an archaebacteria or we have a eubacteria. So meet the archaebacteria. We have many, many different types of archaebacteria, and I'm just going to go through groupings of them. I'm not actually going to go through actual archaebacteria. So we have thermophiles who live at high temperatures. Now thermophiles are going to be in like 135 degrees Celsius water. Those really hot sulfur springs. They're really going to enjoy it. And then we have hyperthermophiles who live at the really, really, really high temperatures. So we have thermophiles. 135 degrees Fahrenheit is going to be like 80-90 degrees Celsius. Now we have the psychrophiles who like it cold. One in the Antarctic grows below 4 degrees Celsius, or best at 4 degrees Celsius, which is really cold. That's below 0 degrees Fahrenheit. Then we have our halophiles who live in very salty environments like the Dead Sea. Acidophiles who live at low pH. pH as low as 1. And remember, pH below 7 is acidic. It goes all the way down to 1. It goes all the way up to 14, and above 7 is basic. So the alkali philes are going to live at high pHs. And then the methanogens, this is what you know, live in swampy marshes and guts of cows. It's what produces methane gas. And that methane gas is, yes, what comes out of that cow. So we're going to classify these bacteria, and what we use to classify bacteria is their shape. We have bacilli, which are rod-shaped, cocci, which are spherical or round, and spirilla, which are curvy. So you can see the bacilli here, the cocci here, and the spirilla over here. And you can see those different shapes. Now, when we name these, we don't just use the spirillae or the cocci or the bacilli. We actually use more. You've heard of Streptococcus. You might have had Streptococcus. Streptococcus is going to be strepto, meaning change, and chains, and coccus, meaning round. So we're going to have long chains of round-shaped bacteria. As you can see here, this would be more like what Streptococcus would be. And then Staphylococcus, or Staphylo, means grape. So it looks like it's just in clusters. And with Staphylococcus, if you've ever heard of someone having a staph infection, they had a staph infection on their arm, something along those lines, they're going to have had that grape-like clusters. And based on the different species of the bacteria, it's going to behave differently. So another thing we need to talk about is gram staining. We have two different types of bacteria here, and a gram stain is going to be able to be the way that we differentiate between these. Now, we have gram-negative and we have gram-positive. All of these cells have cytoplasm, so that's not a big deal. Then we're going to have on the gram-negative side, we have that outer membrane, we have peptidoglycan, and just in a thin layer, and we have that throughout our capsule, and we have it throughout our cell wall. Now, on the gram-positive, we still have the peptidoglycan and the cell membrane, but we don't have that outer membrane, and that outer membrane is what's going to make the difference, and that's going to help us to either determine whether it's gram-positive or gram-negative, because it's going to take up different dyes. So when we gram stain, we gram stain to look for these differences, and in the gram-positive cell, it's easy to take up the purple stain, and the purple stain is going to tell us that it's gram-positive. A gram-negative cell will have a reddish hint to it, so it won't take up that purple stain. It actually takes up a red stain that we use, and so this way we can determine the difference of the layers. So in a gram-positive cell wall, we have thick peptidoglycan. It's about 90% peptidoglycan. It's only one layer. It has small or no capsule. So it's very, very thin, so it's easier to take up this stain. And in a gram-negative cell, we have very thin peptidoglycan. It's only about 5 to 10% here. But we have 3 layers, and we have a capsule around it. And that capsule is going to block that stain from getting in, which is going to make the difference. So a gram-positive bacteria, remember I said, is going to appear purple. Here we have some gram-positive bacteria. And here we have some gram-negative bacteria. Remember I said it would appear red. So let's go on to bacterial DNA. I know I've talked a little bit about it, but I want to get a little more in here. So the chromosome is typically one large circular molecule of DNA. It's free floating in the cytoplasm. Remember, that's the nucleoid region. Now, some have smaller extra chromosomal pieces, and we call those plasmids. And plasmids are something we use in genetic engineering. We take that plasmid and put it into a different organism to get those genes. So bacteria can reproduce themselves in about 20 minutes. So why aren't they taking over the world? They have to have ideal conditions. This is why when we grow bacteria, we put it on a petri dish. It provides nutrients for it. It provides the amount of moisture it needs. It provides the pH it needs. So it provides everything it needs. Also, we have different predators who eat our bacteria. You might not want to eat bacteria, but there are predators who that is what they eat for their energy source. Now, are bacteria good? Yes. There are more good bacteria than bad. Now, bacteria get a bad rap because they're what make us sick. But without these, we wouldn't have nitrogen fixation. And remember the nitrogen cycle? Bacteria can convert nitrogen gas into ammonia, and then other bacteria convert nitrate into nitrate, which we use in our plants. So this is very important. If we don't have nitrogen, we don't have amino acids. Without amino acids, we don't have protein, and without protein, we cannot survive. So here's just a picture of the nitrogen cycle, and just kind of look at it, but we're not really going to go over it. And then bacteria are also good because they decompose dead organisms and waste. Can you remember the other type of organism that decomposes? Good. If you said fungi, you would be correct. Good job. So this allows recycling of organic compounds to be reused by lower members of the food chain. They also help us make foods and medicines, cheeses, yogurts, and antibiotics. Okay, so this is all we have for today, but go ahead and take good notes. We'll go over it in class.